Hi, everyone. I'm going to use this recording to go over the workshop, the elevator pitch workshop that we'll be holding later on this month from 9 to 11. So first, the format of the workshop will be we'll meet together in a room to be determined at 9 o'clock and give each other a brief introduction for each of the trainees, graduate students and postdocs, as well as the faculty members. We'll then go through a first round of elevator pitches. So each of you, there are 6 of you that are enrolled in this workshop so far, will give a 3 to 4 minute pitch to the group and then receive about 3 to 4 minutes worth of feedback from other graduate students and postdocs, as well as your faculty mentors. This feedback will either be usually in the format of they'll give the pitch back to you. What that allows you to do is figure out sort of what stuck, what was well described versus what was a little bit fuzzy in their ability to tell it back to you. And then give you general feedback on areas for development as well as areas of strength. After that first round, each person will go and pair up with their mentor, their faculty mentor, to update the pitch, taking into account the feedback that they had just received from their peers. That'll be for about 20 minutes, and then we'll come back together for a second round of pitches. This will be quite a bit shorter, mostly because a lot of that feedback was incorporated, and so the feedback session will also be shorter, so about 40 minutes, and then we'll wrap up. Okay, so what should your elevator pitch look like? So here is a template or format of the pitch. So you would first want to start off with what is the big picture, sort of what gives the landscape of the biology, such that when you frame your specific problem, it makes sense. This specific problem is often called the micro gap. It could be a gap in knowledge, like we really need to answer this question that has been unanswered for a very long time. Or maybe it's a micro gap in approach or technology that needs to be developed. And then also including a couple sentences on why this remains an open question. This specific problem or micro gap is then followed up by your approach. So what system or set of approaches are you going to use to help solve this problem or help resolve this micro gap? And then a couple sentences on, now that you've used this approach, what's the answer to the specific problem or question that you first generated in step 2? And then where do we go next, whether that's the next key question to ask or the next approach you need or a statement on why you're the best person to answer this question. So feel free to write out your elevator pitch that you'll then deliver live. You don't need any slides or anything. You can just have your piece of paper or whatever in front of you as you're delivering your pitch to the peers. So what I decided to do is give an example of an elevator pitch. And what this will need is I need to identify who my audience is before I deliver my pitch. Okay, so my audience, here is sort of the lay of the land. It's getting harder and harder to get NIH funding. So you're now trying to identify other mechanisms of funding to help support your research ideas. So your friend or your colleague arranges a meeting between you and a venture capitalist. They're really interested in the field of cancer, but without formal training in cancer biology. So you have a 30-minute phone call scheduled. And at the start of the phone call, this venture capitalist starts off the conversation by asking you to tell them a little bit about your research. Okay, so I'm going to go over this. Here is the format of the pitch. And here I'll go over, I think I need to make this a bit smaller so we could see it. The elevator pitch as we go through each step of the format. Okay, so first, outline the big picture phenomenon. Okay. M2 polarized macrophages facilitate cancer cell invasion and intravasation in vitro and in vivo. M2 macrophages promote invadopodium formation, allowing for cancer cells to breach endothelial barriers. Circulating cancer cells seed distant metastatic sites. However, we still don't understand the mechanism of intravasation, which is critical to understanding metastases. So that's outlining the big picture about macrophages and cancer, and then I frame the specific problem. We still don't understand the mechanism of intravasation, which is critical to understanding metastases. Why is this still an open question? Well, intravasation is a rare event to capture in vivo, precluding mechanistic studies examining this process. So what is our approach now to help solve this problem? To overcome this challenge, we have developed organotropic assays to expose the abluminal side of the endothelial layers to cancer cells, better mimicking intravasation. So what's the answer then to this problem? With this system, we discovered that cancer cells can undergo paracellular and transcellular migration through endothelial barriers, and that macrophages induce the formation of invadopodia at sites of cancer cell endothelial cell contact. These results suggest that cancer cells utilize many mechanisms of transmigration to breach endothelial barriers. Okay, so there's the answer. Where are we going to go next? Using our organotropic system, we are now dissecting methods to inhibit cancer cell transmigration. We are uniquely positioned to identify key players of this process in an effort to discover new therapeutic approaches in the clinic. So, I'm talking to a venture capitalist with no formal training in cancer biology, and I bet they're super convinced that my question is the right question to ask and I'm the right person to answer them. Or not. So, what did I do wrong here? Do you think in the way that I have articulated my scientific problem is accessible to people who don't have a cancer biology background or who don't work in my immediate field of research? I think this is really inaccessible. I use a ton of jargon. It's very difficult to understand. And if it's difficult to understand, it's really hard to get excited about the question. And if it's hard to get excited about the question, it'll be really hard to get money to be able to answer this question. So I think the biggest thing about an elevator pitch is you have to know your audience. In this case, I have a goal in mind. I'm trying to convince this venture capitalist that I am asking a really critical question in cancer biology, and that I have a system and I'm the right person to really answer or overcome this challenge. And if I could just get funding, I would be able to do it. And nothing that I have written, even though I followed the exact format of the pitch, allows me to convey any of that to my audience. So how can I revise this such that it's accessible? I hope I could convince them to get excited about it and really make the goal clear. Okay, that's what I have next. Here I try it again. So it's not perfect, but I think it's a lot better than what it was. So some immune cells in your body have a dark side. Instead of helping the host and keeping them healthy, these two-faced immune cells can promote diseases such as cancer. One type of immune cell, macrophages, talk to cancer cells, helping cancer cells move around the body and form a tumor in a vital organ like the heart or the brain. That's the big picture. So let's frame now the specific problem or the microgap. For cancer cells to reach these other organs, the cancer cells often have to enter the blood circulation. And despite the step of entry into the circulation being a vital step in this process, we don't fully understand how cancer cells perform this task, nor whether macrophages help cancer cells perform this task. Okay, why is this still an open question? This is in large part because the outside to inside orientation of the blood vessel is hard to mimic outside of the body. So how have we resolved this problem? We have developed a 3D system that mimics the orientation of entry into the blood vessel, such that cancer cells first encounter the outside of the blood vessel and need to break the vessel to get inside. And what did we find with this system? With this system, we found that macrophages promote cancer cell entry into the vessel and that cancer cells can go in between and through endothelial cells. Because of this unique 3D system, we were able to uncover new mechanisms at much higher detail than previous studies, and we have already identified new ways for how cancer cells get into the blood vessel. So where will we go next? We are excited to continue this project, but we know that we are still missing other cells and environmental components that may be important in the system. So we need more funding to further develop the system so we can identify mechanisms to inhibit this cancer cell movement in an effort to develop new therapeutic strategies in the clinic. So this, where do we go next, is specific to the goals of this elevator pitch, which is to first say, yeah, we have this really unique approach. With this approach, we've already identified new mechanisms of how cancer cells get into the vasculature, but we're not done yet. We need more support so we can continue these studies because we know that we're missing other components. We need to expand out this 3D system further. And this is the reason why I include this in my elevator pitch is because I have remembered who is my target audience. It's a venture capitalist. Now imagine your target audience is, let's say, an undergrad that you're convincing to do a rotation in your lab, or a UROP, work with you in the summer, and they have 4 other labs that they're talking to, and you want to convince this undergrad to do a UROP with you. Maybe what you would include in here is, what are the kinds of skills that you might learn during this UROP? Maybe you know how to do cell culture, but what you don't know how to do is genetics, and I can teach you that. Again, you want to format the pitch for your target audience and with a goal in mind. So please work on your elevator pitch. You could take on this outline. It doesn't need to be exact, but this will allow you to at least have some sort of framework to work with. Try to keep that elevator pitch between 3 and 4 minutes. And we look forward to hearing it and getting your feedback on the other elevator pitches in the session. If you have any questions, feel free to email me. So we'll see you in a couple of weeks.